There are 13 slides, Ma, right?
Yes.
Thank you very much.
I think so, like you said earlier, that the title of the research is Unlocking Nature's Arsenal, the in vitro inhibitory potential of Jatropha tangerensis leaf extract against Plasmodium falciparum. And based on introduction, malaria is a vector-borne infectious disease that is caused by a unicellular parasitic protozoan of the genus Plasmodium. 5 species of Plasmodium cause human malaria, which includes Plasmodium falciparum as the deadliest and the most prevalent on the African continent, with high level of anti-malaria drug resistance. As shown in figure 1, anti-malaria agents usually target the life cycle of the parasite that is characterized by an alternation of generation between sexual reproduction that occurs in the mosquito vector and a sexual reproduction that occurs in the liver and the blood of the human host. Our research is focused on the blood stages of the parasite with a view of finding plant-based treatment alternative through evaluation of Jatropha tangerensis leaf as one of nature's cheaply available food. Considering the high cost of synthetic drugs, it is high time we start consuming food as medicine rather than consuming medicine as food. Based on our specific objectives, number 1 is to culture and maintain clinical isolates of Plasmodium falciparum. The second objective was to determine the in vitro susceptibility pattern of the Plasmodium falciparum isolate to Artemata. And lastly, to evaluate the in vitro anti-plasmodial activity of Jatropha tangerensis leaf extract against Plasmodium falciparum isolate at the early and late trophozoite stages of the intra-erythrocytic cycle. Plate 1 shows images of the materials and methods that were taken during the laboratory work, where 10 malaria parasite positive samples were collected from the Ahmadu Bello University Medical Center, Zaria, and 4 different extracts were collected from pharmacology department, pharmacognosy department of Faculty of Pharmaceutical Sciences, ABU Zaria. Plate 2 shows the photomicrograph of the confirmed Plasmodium falciparum erythrocytic stages, which indicates the double nucleated early trophozoite, the single nucleated early trophozoite stage, the macrogametophyte form, and the alcohol form. Plate 3 also shows another photomicrograph of the confirmed Plasmodium falciparum erythrocytic stage. Results and discussion. Figure 2, which is indicating the growth rate of Plasmodium falciparum in vitro culture. The result shows that the malaria parasite sample 1 to 9 have indicated increase in percentage parastemia, which range from 0.5% up to 4.3% after the culture. However, malaria parasite sample 9 did not indicate any increase in percentage parastemia after the culture, which may be that the sample was collected from a patient that have already started anti-malaria treatment before the hospital visit, which has made the parasite vulnerable before the culture. And our finding here is supported by the findings of BASCO 2023, who reported that the growth rate of Plasmodium falciparum and the ability of it to survive in vitro culture varies depending on the initial parastemia, the extent of red cell dilution, the quality of the serum, as well as recent exposure to drug treatment before cultivation. Trader and Jensen in 1977 also added that the major factor that is responsible for this variation could be the undefined innate characteristics of the host erythrocyte to support the parasite growth. Figure 3 shows the susceptibility pattern of Plasmodium falciparum isolates to Artemata, where the Plasmodium falciparum isolates 1 to 10 have indicated IC50 values in micromole per liter, which were all below the peak plasma concentration of Artemata, which is 1.81 micromole per liter. However, Plasmodium falciparum isolate 4 and Plasmodium falciparum isolate 8 had the highest IC50 values, which is approximately 1 micromole per liter. This hints the possibility of this isolate for future resistance to Artemata, and thus they are selected for further studies with the extract. So our findings here is aligned with the recent findings of Handbook Pakan and White 2022, who reported that artemether is nearly as active as artesunate in the treatment of malaria, although artemether is slowly absorbed. Being an oil-based injection, it is slowly absorbed in body, while compared to the water-soluble artesunate that is rapidly absorbed. However, the findings that indicates complete susceptibility of the isolates to artemether is confirming to us that hitherto artemether is still efficacious in the treatment of malaria. Figure 4 shows the antiplasmodial activity of extracts of Jatropha tangerensis leaf against Plasmodium falciparum isolates at the early trophozoite stage, where results indicate IC50 values that are significant at p-value less than 0.05. From the results, we could see that the sequential ethyl acetate extract had the highest antiplasmodial activity, being that it recorded the lowest IC50 value in microgram per mil compared to the other extract. And when compared to the positive control, we could see that the positive control artemether, lumefantrine, and quinine have recorded higher antiplasmodial activity than the sequential ethyl acetate extract. However, there is an indication of reduced susceptibility of the isolates to artemether lumefantrine, which also coincides.
I'm sorry, Ma. I'm sorry, but
which
the time.
Sorry.
Time,
Pardon me,
please.
please.
All right.
Okay,
all
let
right.
me just round up.
Thank you.
Let me just round up the presentation.
Thank you.
So this is just indicating that at both stages, the early and late trophozoite stages, the sequential ethyl acetate extract of Jatropha tangerensis leaf has elicited the highest in vitro antiplasmodial activity against the early and late trophozoite stages of Plasmodium falciparum, indicating that it has the potential of becoming a future anti-malaria treatment alternative, since it had an IC50 value of 4.77 microgram per mil, which is a range to up to 5.83 microgram per mil. So our recommendation is that although the synchronicity of Plasmodium falciparum in intra-erythrocytic stages in culture system is easily lost, which is one of the challenges that we had. We couldn't get up to the last stage of the erythrocytic cycle. More studies on antiplasmodial activity of Jatropha tangerensis leaf against all the intra-erythrocytic stages of Plasmodium falciparum should be conducted in order to fully understand the mechanism of antiplasmodial activity. Thank you for listening.
Thank you very much, Mark, for this wonderful presentation. We now have 3 minutes to entertain comments and questions the members in attendance. So if you have any question, kindly. Well, I think in the absence of any question, we will move on to the next presenter. Thank you very much once again, Mark.
You're welcome. Thank you.
Tahiru.
Yes, I was trying to share the slide to you.
Yes, I saw it. It's like there are 16 slides.
Yes, sir.
I hope you can see the slides. Dr. Atayu, please, can you present it?
Yes, sir.
I think everybody can see.
Yeah. Good morning to you all, my colleagues and all the presenters. My name is Bashar Atahiru from Kebbi State University of Science and Technology, Arewa. I was presenting on molecular identification of hepatitis B infection amongst pregnant women attending antenatal care at General Hospital between Ziga and Arewa. I can say about the introduction hepatitis B. The word hepatitis B has derived from Greek word. Hepa means liver. Itis simply means inflammation. Hepatitis B means the inflammation of the liver that caused by the virus. However, it may be caused by certain autoimmune diseases or bacterial infection. It can cause chronic infection and put millions of people at higher risk. Hepatitis B virus lives in the blood, other bodily fluids such as semen, vaginal fluids, and others. Then it can be transmitted from one person to person through unprotected sexual intercourse or an uninfected person sharing of forms, infected sharp objects. And the virus has been detected from peripheral mononuclear cells, tissues of pancreas, spleen, kidney, and skin, and fluid of saliva, semen, sweat, breast milk, tears, urine, vaginal secretion, and human urine. Hepatitis B is an inflammation of the liver caused by the hepatitis B virus within the family of Hepatinidae. The condition can be self-threatening or can progress to fibrosis, scarring of the liver cancer. Hepatitis B virus are the most common cause of hepatitis B in the world. But other infection, toxic substances, alcohol, certain drugs, and autoimmune diseases can also cause hepatitis B. Hepatitis B is highly infectious. Some are saying it is 50 to 100%, 50 to 100 times more infectious than HIV. So that's how dangerous the hepatitis B virus is. Hepatitis B viral infection is a global public health problem. The justification the hepatitis B viral infection is a major public health problem, significant cause of morbidity and mortality nationwide. People usually have a higher risk of hepatitis B virus infection due to immune resistance and improper blood screening before donation, which is a result of inadequate awareness about the hepatitis B virus infection. The screening of pregnant women is an essential step in reducing the global burden of chronic hepatitis B infection. Therefore, studies from different settings are crucial to reduce the burden and the problem to make informed decisions. And the aim of this study is to assess the prevalence of hepatitis B virus infection among pregnant women attending antenatal care, general hospital, Jiga, and early metropolis. While the aims of this study is to remind the prevalence of hepatitis B infection amongst pregnant women in the study area, determine the risk factors associated with the virus during pregnancy, to identify the hepatitis B infection using serological tests, ID like strips or one-step rapid diagnostic tests, to detect hepatitis B virus using polymerase chain reaction (PCR) method. And the study was conducted between Jega and Aleru, which with an area of Kebbi State, northwest geographical zone of Nigeria between the longitude and longitude of the state made of 21 local government and so on. Then the majority of which are civil servants, traders, farmers, teachers, and students, Kebbi State. And sample size, the formula used for sample size is n is equal to z square p 1 minus p divided by, where n is number of sample size, while z is standard normal derivative at 95% confidence interval. Prevalence from previous study is 8%. D is allow margin of errors. Then sample collection, approximately 2 ml of all blood were collected from each consented pregnant woman that wanted to participate in the study. Those that don't wanted to participate compound verbally. Then the 2 ml of blood were collected from 113 consented pregnant with the help of registered lab personnel and put into a plain container with a white-mouthed cover. To do the following procedure, the patient was asked to sit comfortably gently. Tourniquet was applied on the upper arm. After cleaning the anticoagulant fossa, the needle was introduced into the vein to withdraw the blood. After the needle was introduced and withdraw the blood, the cotton wool was applied to stop the bleeding. Then sample processing. The blood were transformed into the test tube containing EDTA, which is ethylene ditetracetic acid. Then the immersion into the serum for 5 seconds. The strip was placed on a horizontal position on a non-absorbent surface for 15 minutes, after which the results will be read and recorded. Sample. Positive sample generates double red color, both at the band test and control region, while negative sample generates a red color band only at the control region. Then PCR detection of hepatitis B infection. Volumetric chain reaction was done using Topa polycyclate reaction mixture. Then we move to the results. The distribution of hepatitis B infection according to
hello, doctor.
Hello.
I'm sorry.
Yes.
I'm sorry to interrupt. Please, time. Please consider the time. Maybe it will be better. Summarize.
Okay.
The time allocated has already passed. Thank you.
Okay, sir. I know. Conclude. Let me just do conclusion. I already moved to the result. Considering the overall prevalence system, we concluded that Jigan Ali local government has a prevalence of 15, which is 8.5%, 98, which is 86.7%. Patients were negative. Since the virus can be transmitted from infection mother to the offspring, especially adverse, the presence of hepatitis B infection in women in general and pregnant women in particular calls for concern from the government and other agencies to know the burden and how dangerous this hepatitis B virus is in the locality, in the country, or in the state in general. Thank you for listening, sir.
Thank you very much, Dr. Participants, the floor is now open for questions and comments. Dr. Atahiru, please, maybe before the participants' comments, can you hear me?
Yes, sir.
Thank you. I want to ask a question regarding your prevalence that you have of 8.5%.
Yes, sir.
That prevalence,
You see,
other prevalence. I said if we compare the prevalence you obtained with other prevalence values reported in other studies, is it high or low? Does it call for concern?
Sir, it's called for concern because due to the burden of cost, we need to know that this virus is dangerous in this particular area and the locality. So whether it's low or high, it needs for concern. That's from my own perspective, according to what I see.
All right, all right. So participants, please, anybody else has a question for Dr. Tahiru? If not, I think we would be considering moving to the next one. Hello, participants. Any question for Dr. Tahiru? If we don't have any, we are going to move on to Malachi. Malachi, please, if you are, it's like you are muting. You are muting your microphone. Please, if you are ready to present, kindly share your slides.
Can you see my screen?
Yes, you can see your screen. That's wonderful. Please, can you share your slides?
So, can I start?
Yes, but please, we would like to see your slides. Thank you very much, sir. You have 7 minutes to present your research.
Okay. My name is Osuji Malaki Ikoku. The title of my presentation is Use of Different Substrates in Biogas Optimization Process and Their Comparative Effect. I am co-presenting with Obulie J.N., Nweke C.O., Wanyawu C.E., and Udogu C.V. First of all, before we start, I would like to let us know what biogas is. Biogas is a mixture of gases produced during the anaerobic digestion of biological or organic materials, and it takes place inside a biodigester. That will take us to know what is biodigester. It's just like a system that promotes decomposition of organic matter. It produces biogas generated through the process of anaerobic digestion. That anaerobic digestion has three processes. The first one is called hydrolysis. The second one is acid production, or what we call acidolysis. And the third one is acetate production. And the final one is the methane production. Materials and methods. The sample used in this research or investigation include cow dung, poultry, human feces, and the one from piggery. The cow dung and the poultry dung were collected from a place we call Obis, a livestock market along Owerri Port Harcourt Road. The other substrate, that's the piggery and the human feces, were collected from private homes. These four substrates were transported to the project lab for anaerobic digestion. 2 kilograms of each of the substrates was collected from the sample site. Then I went ahead to fabricate the anaerobic digester using plastic containers of 20-liter capacity, and they were of the same size, quality, and transparent. The four digesters, they are all airtight. I'm talking of the cover. And I used an electric drill to bore a hole on the cork and inserted a bronze nozzle and used a glue to seal it so that it will be airtight. And I had to attach a transparent hose that is connected to a tube. You can see how I fabricated the digester. You can see how transparent hose was connected to it. The slurry formation, that slurry is those substrates, you then mix them with water. Separate buckets were used to prepare the slurry by mixing 1 kg of each substrate with 9 liters of water. After thorough and homogeneous mixture was achieved, they were each poured into the designated fabricated digester and labeled appropriately. This is the buckets I used in mixing the slurry separately. Then the anaerobic fermentation and gas production. The fabricated digesters were placed on high elevated flat surface, and the tube were placed at about 3 feet below. This is the shear downward gas delivery. The type of fermentation process used here is batch culture fermentation, because after pouring the slurry into the fermenter, you seal it, you will not open it again until after 15 days. And I kept on monitoring it at interval of 3 days. This is how I placed the digester on top and the tube connected with a hose for each. During the process, I did physicochemical analysis of the substrate, that is the total solids, volatile and fixed solid, moisture content determination, and the pH of the substrates. Results: after 15 days of fermentation, what we call retention time, these results were obtained, but initially between the 0 and the 3, it was as if nothing is coming out. And though I cannot categorically say it is the lag phase of but I know the indigenous microorganisms were trying to get themselves acquainted and to start the digestion process. So this is the table of what I got after 15 days. The digester 1, which has human feces, the day 1, that is 0, that is the weight of the tube. The day 3, after weighing it and subtracting it from the weight of the tube, I got 0.10 kg. Day 6, 0.07 kg. Day 9, 0.80 kg, like that until day 15. Then to the cow dung, day 3, 0.70 kg. The same thing day 6, the same thing day 9. To the piggery, the same thing you can see from the slide. And then to the digester 4, which has poultry dung. You can see from it. And now try to represent them in graphical form. But from this is for human feces. This one is for cow dung. This one is for the piggery. And this one is for the poultry dung. Let me try to interpret the results. The results of this research were obtained using electronic weighing balance and recording accordingly. The weight of the tubes we are taking the first day, and subsequently we are subtracted from the weight at 3-day interval. The table above shows the results of the weight of the tube at interval minus the weight of the tube before. It was also observed that the quantity of gas started changing from day 3, which means from day 0 to day 2, nothing was happening until from day 3 until day 15. You started seeing that the tube, the tube started increasing, which means gas has started entering into it. Like I said, yeah, it is scientifically difficult to say exactly at which interval that corresponds to hydrolysis, acidogenesis, acetogenesis, and the methanogenesis. But the knowledge of bacterial growth curve explains why the gas production did not start instantly. That is what we call lag phase in bacterial growth curve. At that lag phase, you hardly notice any change because the bacteria, they are just trying to get themselves acquainted with the substrate and the fermentation process. This could be attributed to bacteria passing through the lag phase until log phase, where you start seeing significant changes. Also, similar research done by Ikoku et al. 2023 showed that keeping the digesters inside would have great impact and effect on biogas production. This could be attributed to the effect of temperature because I kept the digesters inside. But similar research showed that when you keep the digester outside, you have enough gas because the temperature, the environmental temperature, as the sun is touching the digesters, the reaction and the fermentation process will increase, leading to production of more gas. Leading to the production of more gas. Conclusion and the recommendation: biogas formation can be achieved using locally sourced substrates or biomass. In this research, comparison of the different substrates, human waste, cow, and poultry, we are compared. The digester 4, which was for poultry dungs, gave the highest yield and is recommended for biogas production. Thank you for listening.
Thank you very much, Dr. Ikyoku, for this wonderful presentation. We now open the floor to participants for questions and comments. Meanwhile, before the participants ask questions, Dr. Ikyoku, if you can hear me, I'd like you to shed more light on 2 things.
Okay.
Number 1, in the process of your research, you did physicochemical analysis of the biogas substrate.
Yeah.
So please, how do we relate the physicochemical parameters and the yield of the biogas? That is 1. And number 2, you stated that, or based on your results, the piggery substrate has the highest yield.
No, poultry.
Okay, poultry. Yes. So please, is there what does that indicate? Is it the difference in the carbon to nitrogen ratio of the substrate, or we want you to shed more light on that, please? Thank you.
Yes. From your last question, poultry dung gave the highest because of the, like you said, carbon to nitrogen, and even though all of them, they have nitrogenous content, but that of poultry, the nitrogenous content is higher. the nitrogenous content is higher, and even when you feel it, when you bring them out, if you feel it, you discover that that of poultry dung is even hotter. It's even hotter compared to other ones. Other ones, naturally, when you touch them, if you try to touch them, maybe you're wearing hand gloves, they are somehow cold. But that of poultry is hotter, which means ordinarily, before you even mix it with water, the reaction have started taking place, the fermentation have started taking place. It's hotter, and the nitrogenous content of that poultry waste is higher compared to others. That of human, the nitrogenous waste there is small. That was why it didn't give enough for biogas. If you look at the percentage, you will see that the human waste gave 18.86%. Cow gave 20.75%. Piggery gave 22.64%, and poultry gave 37.73%. And another research which I did sometime when I what we call co-digestion, I used poultry and pig in similar research. In fact, the 6th day, the tube busted. I had to go and look for a bigger tube, which means I will also recommend, if somebody wants to go into biogas production and you are looking for substrates, go for piggery and that of poultry. They will give you enough when that what we call co-digestion, combining, or if you like, even if you use cow, poultry, and piggery, they will give you a perfect and enough production of gas.
Thank you for the explanation, sir. Please, if you have any question from the participants, feel free to ask. If not, we'll be considering other presenters. Thank you once again, Dr. Malaki, for this presentation. We don't know if you're welcome. We don't know if Shafi'u is available. He has been having technical issues. In his absence, Salih Ibrahim is here, but it's like he's muted. Salih, please, if you can hear. Hello.
No, I'm not. It's not Salih who is. Saadat Lawal. I'm present.
So maybe Ma would allow you to present before Salih gets ready. So your slides, Ma, are they ready?
I'm not. I've been trying to share them, but I don't know what's happening. My computer is not connecting with my phone to share them, so I don't know if I can still present without sharing them.
It's all right. It's not a problem. We may allow you to present without the slides.
It's actually a review paper. It's not actually an experimental. It's a review paper that I wrote on mycosis in a changing world, climate-related issues in a fungal infection with. My name is Lawal.
It's all right.
Hello.
Hello. You may continue, Ma.
Yes,
7 minutes.
My name is Dr. Saadat Lawal Bagiwa, and I'm presenting a paper on mycosis in a changing world. Can I continue?
Yes, you continue. We are hearing you.
Okay. Mycosis in a changing world, climate-related issues and fungal infections. So fungal infections are known as mycosis, and climate change affects prevalence and distribution of fungal infections. It has become one of the most important global challenges in this era, with detrimental effects on the ecosystem, human health, and biodiversity. So this paper tried to look at the interconnectivity between environmental factors, fungal pathogens, and human health. And so some of the risk factors for this are geographic, as a result of geographic expansion, which leads to changes in temperature and precipitation, breeding fungal habitat. E.g., for example, the fungus that causes valley fever, which is coccidioidosis, has expanded its range from traditional Sahara desert areas to coastal regions. Then, of course, there's also the emergence of new pathogens through adaptation to changing environmental factors, allowing them to colonize new hosts and ecosystems. And there's also the problem of population movement, which also affects climate change as a result of climate change, which may contribute to population displacement creating crowded living conditions which facilitate fungal spread. And also extreme weather events like storms and wildfires can disperse fungal spores, leading to introduction of new pathogens to vulnerable communities. And there's also selective pressure through higher temperature and UV radiation, which may promote the growth of some virulent fungal strains, raising health concerns. And environmental disruptions can also aerosolize fungi, facilitating their spread. And some of the symptoms of this are not easily detectable because they might mimic other infections such as pneumonia, which are bacterial. And the common symptoms, of course, include cough, fatigue, night sweat, fever, and the rest. This might lead to misdiagnosis and incorrect treatment are possible. Some common mycosis include valley fever, histoplasmosis, blastomycosis, and aspergillosis. So fungi can cause devastating mortality, morbidity, and economic loss, impacting agri and food supply chains. The interconnection between people, animals, and the environment is crucial to understanding fungal disease. Some might be zoonotic, while others can affect both humans and animals. So we can prevent this it's mostly preventative anyway. So you can maintain hygiene, avoid overcrowding, be aware and alert of what is happening regarding the symptoms, and follow medical advice. And also reduce exposure to extreme weather events such as flood and storms, which can disperse the pathogens that cause these diseases. And it's also in line to support policies aimed at reducing greenhouse gas emissions, which lead to all this destabilization and lead to emergence of new fungal strains. And there's also a need for collaborative efforts to access disciplines to address the complex challenges posed by fungal changes. By recognizing the interconnectivity, we can enhance our ability to prevent, treatment, and manage fungal infections effectively. So I hope I've presented what I tried to write something that I not get the slides. So
Thank you very much.
Yes, I hope you're able to understand what I'm trying to portray here.
We were able to understand you more. So thank you for the presentation. The floor is now open for comments and questions to Dr. Saadatu, based on her presentation. In the meantime, as we wait for questions from the participants, I'd also like to ask for a clarification, Doctor, regarding global warming in particular. Yes, what is the direct link that we can establish between global warming and the spread of fungal mycosis?
Yes, very good. So thank you. You know, global warming generally leads to rises in temperature in the environment. And it causes different types of precipitation at the wrong time. Sometimes there's rain when you're not even expecting rain. Different erratic weather conditions occur. So this might actually cause this fungi to actually adapt and change their environment. Sometimes you can find fungi that you normally find maybe around the desert area. Because of all these changes, it might actually move to the coastal areas where it was not actually expected to be there. Then it will it might actually develop and become a more virulent strain, leading to new types of infections that are not easily detectable.
I understand. Any question from other participants? In the absence of questions, we thank you once again, Doctor, and we move to the next presenter.